introduce Professor Tuor. Professor James Tuor is a TT and W.F. Chow Professor of Chemistry at Rice University, United States. He obtained his PhD in chemistry from Purdue University and then pursued his postdoctoral research at the University of Wisconsin and Stanford University. His research interests include carbon materials chemistry, molecular electronics, synthetic organic chemistry, and nanotechnology. He's a highly cited researcher with over 715 research publications in internationally renowned journals and over 140 patent families with an H index of 150. He's also well known for his perspectives on on the molecules needed for life. He also received widespread international recognition, to mention a few: the Royal Society of Chemistry's Centenary Prize for Innovation in Materials Chemistry, the Trotter Prize, the Finman Prize in Nanotechnology, the NASA Space Act Award, among many other awards. Professor Tuor was ranked one of the top 10 chemists in the world over the past decade. So it gives me great pleasure to invite Professor James Tuor to make his presentation, please. Professor Tuor.
Okay, well, good to join you here. This is early morning for me, but let me go ahead and bring up my presentation here, and we'll get started. Okay, so I thank you for this invitation, and I'll give you some of my thoughts on a critique of the current origin of life research and where we are. Let me just start by showing you my family. What I really want to point out here is my wife, Shireen, right here. She's a Punjabi. So we've been married, we are now in our 40th year of marriage. So we've been married for almost 40 years, 39 years plus. And we have 4 children and 2 grandchildren. And you can tell that this is the one who's not genetically connected. That's my grandson, my son-in-law. And but I always feel like when I speak to people from that part of the world that I'm somehow connected through my lovely wife. Okay, I'll give you just kind of a background overview on the things that I do, and then we'll get into my critique on the origin of life. So we work on an area called laser-induced graphene, which is turning any carbon substance into graphene by hitting it with a laser. You can even do it with a piece of bread or a coconut and make a supercapacitor out of coconut. We split carbon nanotubes to make graphene nanoribbons. I'll show you on the next slide what we can do with those. We've made, and this is now sold by Millipore Sigma Aldrich. We've made electronic memory, which is transparent. This is now in a commercial company. It's a public company called Weebit. This is, we're working on traumatic brain injury and stroke. We do a lot with batteries. This is carbon nanotubes that are melded directly into graphene. And there's a battery company that we've started called Zeta. We work in the area of carbon quantum dots that we make from coal. We've learned how to take waste plastic and convert it into material that can absorb carbon dioxide. We've started another company recently where we can take any carbon material and convert it in about 10 milliseconds into graphene. And this is now making bulk graphene. And so that's in a new company called Universal Matter. We've learned how to 3D print graphene. We can take single molecule nanocars. We build in these Faringa motors, shine light on them, and they will turn, and this car will move along along a surface. We can take the same motors and we can put a peptide on them, have them recognize on a particular cell surface, drill into that cell and kill the cell. So we're using these in medicinal applications, and this is where you've destroyed the cell wall of a superbacterium. This is a rat which has had its spinal cord completely cut in two at C5 at the base of the neck, completely cut in two. And then we put 1 drop of a polyethylene glycol solution, 1% of the graphene nanoribbons. That will then start bridging this gap, and then the connections reform, and the rat can get up and start walking 2 weeks after surgery. And so that was with a completely cut in two spinal cord. Scored an 18 out of 21 on a mobility scale. 21 is optimal mobility. And then after 3 weeks, you'll see this transition at 3 weeks, scored a 19 out of 21. And so she's able to run at this point right after 3 weeks. So these are some of the things that we're able to do with these graphene nanoribbons. So we've started a number of different companies in the past 6 years, these 14 different companies, from everything from medicine to materials and electronic memory. And then we have a bunch of other companies in the waiting that are going to form this year and next year. So we try to take what we do and really bring it forward. Okay, with that, let's talk about abiogenesis. All right, abiogenesis, the origin of life from non-living matter. To construct any convincing theory of abiogenesis, we must take into account the condition of the Earth about 4 billion years ago. These are not even my definitions. I'm taking this right off the web. So for synthesis to be categorized as being prebiotically relevant, it must use chemical reagents and conditions that are presumed to be available on an early Earth. Abiogenesis takes place before biology and before biological evolution can begin. Hence, it's prebiotic or prebiology. I am fine with these, with life having been seeded upon Earth, or these molecules that I'm going to talk about having been made some other place and then coming to Earth. It doesn't matter. We're talking about the origin of first life. How did that first life form? All right, what are the characteristics of life? You have to have responsiveness to the environment, growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis, and being made of cells and passing traits on to offspring. Again, that is a standard textbook definition of the characteristics of life. Homeostasis is a steady internal physical and chemical condition. All right, so you have to be able to have growth and change, because now what people are trying to do is to redefine life into their own image of what they want it to be so that they can try to make it. But this is what we've always known as life. It has to be made of cells, has to have a metabolism, has to be able to breathe, maintain homeostasis, passing on traits to offspring, has to be able to reproduce, have growth and change, and be responsive in the environment. Molecules don't care about life. Organisms care about life. Chemistry, on the contrary, is utterly indifferent to life. Without a biologically derived entity, like a person acting upon them, molecules have never been shown to evolve toward life. Never. Just doesn't happen. Almost every chemical synthesis experiment in origin of life research can be summed up by a protocol analogous to this. You purchase some chemicals, generally in high purity, from a chemical company. You mix those together in water at high concentrations in a specific order. You obtain a mixture of compounds that have a resemblance to one or more of the basic 4 classes of chemicals needed for life, which is carbohydrates, nucleic acids, amino acids, or lipids. Publish a paper making bold assertions on these compounds that are usually stereo-scrambled, engage with an ever-gullible press, and they turn up the knob of unjustified extrapolations, and then watch the mesmerized layperson say, you see, scientists understand how life formed. And then you encourage a generation of science textbook writers to make colorful, deceptive cartoons of raw chemicals assembling into cells, which then emerge as slithering creatures from a prehistoric pond. Molecules that compose living systems almost always show homochirality. When building molecular systems, constant redesigns are needed, which take the synthesis right back to step 1. It's often impossible to remove a moiety once you've put it on the molecule. Synthesis reactions don't know how to stop their current course of progression or why to stop. There's no target. There's no goal. They don't know what they're going toward. Time, although claimed to be the great savior of abiogenesis, can actually be the enemy. For example, carbohydrates are kinetic products. They undergo caramelization. They undergo the Cannizzaro reaction. A prebiotic system does not have the ability to easily purify the structures. Reagent addition order is essential. The parameters of temperature, pressure, solvent, light or no light, pH, atmospheric gases or no gases have to be carefully controlled. The characterization at each step is essential for the chemist, but hard in a prebiotic system. Mass transfer. If any of you have ever done organic synthesis, multi-step organic synthesis, you know what it's like. You start with a material, you might start with a kilogram of material. After 20 steps, you're left with a milligram of material. So very often you're going back and bringing up more material from the rear. How do you do this when nature doesn't keep a laboratory notebook, so it doesn't know how to go back? It doesn't know how to go back. It doesn't know what it's going toward, nor does it know to go back to make more of what it made. So carbohydrates are the same as sugars, which are sometimes called saccharides, and the polymers therefrom. All right, so here's some naturally occurring monosaccharides. There's a bunch of them. And then you can have homopolysaccharides, so you hook just these same ones together. Now, a lot of saccharides are not homopolysaccharides. A lot of them are not. And so even table sugar is not. So to suggest that they're all mostly homopolysaccharides is inaccurate. But even if you have homopolysaccharides, you can have the beta 1,4, the alpha 1,4. You can have alpha and beta linkages. You can have 1,4, 1,6. Lots of different ways you can hook these things together. It's a big problem. You have all of these different sugars you have to deal with. And so how do you choose? Which one are you going to use? If you're going to make RNA or DNA, you've got to have ribose. And so you have to have the ribose or the deoxyribose. But then what you've got to be able to do is even on this very simple little system, you've got all of these 8 isomers. These just from the pentose sugars. If you're making a saccharide, if making the saccharide wasn't hard enough, how do you control those stereocenters? Hooking them together is just as hard. So here's D-glucose. Here's D-glucose, and there's lots of different ways that D-glucose can hook together. So you can hook it together from this OH to that one, or you can hook it up from this OH to that one. And all of these are different. And you can have some of being the 5-member ring. So glucose can be in the open form with 4 stereogenic centers. It can be in the closed form in a 6-member ring with 5 stereogenic centers. It can be in a 5-member ring, again, with 1, 2, 3, 4, 5 stereogenic centers. So each of these has 5 stereogenic centers. So each has 32 possible stereoisomers for each of these forms. So that's a lot. Now you think of hooking this together. It's a problem. So polysaccharides, you have to be able to connect these. This is showing the beta 1,4 glycosidic linkage. And so you can have these sorts of glycosidic linkages, or it could have been in the down position. So this is cellulose. And so you can have starch. Starch can have the alpha linkage. So starch has this alpha linkage, and that's why we can actually digest this. We don't normally eat grass. We don't eat cellulose for nourishment. We might eat it for fiber, but we don't eat it for nourishment. This is something we can digest. And so starch is very similar, but it has just a slightly different linkage here. Now, if you take, for example, D-glucose, just 6 D-glucoses, how many ways can you hook these 6 rings together? How many ways? Well, it turns out you can hook these together more than 1 trillion different ways. That's amazing. So if you think of DNA, say you had the DNA with an A base on there, what's the number of ways that you can hook those 6 DNA, those 6 nucleotides together? There's only one way. It'd be A. That's it. Well, how many ways can you hook just D-glucose together? Over 1 trillion different ways. And if it's not hooked together right, it doesn't work. Just 6 units over 1 trillion different ways. Nobody knows how this thing was hooked together. Nobody knows. Very, very difficult to figure this thing out. Enzymes do it, but there's no chemical methods that we could ever fathom to do this. The way we do this chemically is you have to block all the hydroxyls that you don't want connecting. So again, this is very difficult chemistry. So summary, carbohydrate synthesis is exceedingly hard. Coupling of carbohydrates together is painstaking. The untrained have no idea how hard the syntheses are to perform. The trained remain clueless on how these could have formed in a prebiotic system. The untrained see little difficulty in conducting these syntheses. All right, peptides, proteins, amino acids. That's the next class of compounds. So you have 20 common naturally occurring amino acids. Some of them have fairly simple side chains. So 19 of them have a stereogenic center, have one stereogenic center, at least one stereogenic center. And so you see that some can have, but others can have more reactive side chains, amino acids with reactive side chains. These themselves can react. Then you can have other amino acids, again, with reactive side chains. This one, this one, this one, with reactive side chains. Again, these all have reactive side chains. Again, these all have reactive side chains. So the problem is, how do you get these things hooked together? Because amino acids don't just hook together. You've got to keep those active side chains protected. And this is how we do it chemically in a laboratory. This is how it's done. You have to do this. SPG means side chain protecting group. You got to do this. You can't make amino acids with this. Amino acids don't spontaneously polymerize in water. They actually depolymerize in water. Now, if you want to say that they were in some sort of hydrophobic gel, okay, but how did you deal with the side group protection? We have no solution to that. What I'm showing you is that at each step, there's a problem here. All right, even origin of life researchers themselves are saying it is inconceivable that these sophisticated and coordinated macromolecules suddenly emerged at the origin of life. In practice, there are good reasons why these reactions are ineffective in water, because they're zwitterionic. They don't go in water. All right, nucleotides, DNA, RNA. So nucleotides, so you have your ribose, and then you have your base, ribose and base. So DNA will have predominantly 4 different bases here based on a deoxyribose, and RNA will have 4 different bases here based on a ribose. So the ribose, so for DNA, you're just pulling this one hydroxyl off, and you have deoxyribose. Oh, by the way, we don't know how to pull that off. I mean, it's not something that just falls off. So it's just tough chemistry to do. The way we deal with these now is we buy them from manufacturers who isolate them from natural sources. All right, so solid phase DNA synthesis. So what you have to do, this is what's involved in a solid phase synthesis. You have to keep all of these other groups blocked. And then you have to have activation here to get the couplings to go. You have to block these ones that didn't grow, cap them off, and bring it all through these oxidation steps. This is not easy. So a lot of synthesis here. The other thing about it is RNA is unstable. RNA is unstable. Even if you look on the websites on where it is sold, they will tell you RNA is much more unstable than DNA due to its chemical structure. RNA is subject to autocatalysis, meaning that hydroxyl can hit that phosphate and it breaks the bond. And so it leads to autocatalytic degradation. So it is stored with a tris buffer. So it is stored with this chelating buffer to keep these metal ions out, because that promotes this even further. It has to be stored at minus 80 degrees centigrade. So all these models of these things floating around at room temperature and the edge of a volcano, this is nonsense. They don't stay around. It just goes back. So it it goes back. It just goes to monomers. Doesn't stay as RNA. RNA is unstable. So your whole RNA world hypothesis is dealing with a material that's quite unstable. Lipids in the cell membrane. So the next class of molecules you have to deal with are the lipids. So what lipids are is they'll have these 2 hydrophobic chains, and then they'll have this ethanolamine on this phosphate, and you have a stereogenic center there. So all of these compounds I've showed you have all these stereogenic centers. Even lipids, which you think are simple, have stereogenic centers. Now, these are simple lipids. There's other more complex lipids like sphingolipids that have 2 stereogenic centers or 4 possible isomers. So the structure of phospholipids. So you form this lipid bilayer where you have the hydrophobic portion coming here, the hydrophilic on top, hydrophilic and hydrophobic going in. All right, so there's the summary to address the origin of life research. There's no control of absolute stereochemistry. You have to have homochirality. There's little to no control of relative stereochemistry. There's little to no accounting of yield. There's little to no accounting of purification. No accounting for mass transport through the sequence. There's enormous human intervention in every phase of the experiment, which you wouldn't have on an early Earth. Prebiotic Earth would have none of these luxuries. The origin of life researchers, those superb synthetic chemists, are generally silent on these essential synthetic questions. If you don't purify, it's going to gum up the works. It's not going to work very well. You have to account for high yields. You have to account for the relative stereochemistry, the absolute stereochemistry. When you read articles by origin of life researchers, it leaves enormous holes which render their work prebiotically irrelevant and misleading to the non-synthetic chemist. This is not for lipids only. It is for almost all origin of life researchers' syntheses. Chiral-induced spin selectivity. This is another problem that we have to deal with. So some people say, well, chirality came later. Well, did it really? Well, we're learning about what's really important for these reactions to go. So what's known? In nature, you can have two hydroxyl radicals form hydrogen peroxide by going anti-parallel down a singlet surface. So if you have one of these spin up, the other one spin down, they're anti-parallel, they go across a singlet surface and they form hydrogen peroxide. If they're parallel, if those two electrons are parallel, they will undergo a triplet surface and they form oxygen, O2, and two protons. These unpaired electrons. So how does this happen? How does nature cause hydroxyl radical to go in one of two ways depending on how it wants it? It actually uses what's called chiral-induced spin selectivity. And so what happens is the electrons, that it turns out that these chiral molecules are spin valves. And they will select out only the electrons that have the right spin to go down this pathway. So electron transfer in cellular systems, photosynthesis, for example. Plants survive by photosynthesis, which have relatively long-range electron transfers. When traversing down the proper direction, meaning when the spin and the chirality of the molecular architecture are matched, the electron backscattering by vibrational modes is suppressed, and then you get this reaction, because they suppress this change, which would demand a change in linear momentum. Hence, inelastic collisions and energy loss as heat are suppressed. Many people have wondered, why is it that cells don't just burn up with all the amazing chemistry that's going on? And this is why, because they're spin valves. They use the chirality to promote reactions. What we have taught our students for years, and this is the way that I was taught, that the reactions in nature go because there are enzymes with a chiral cleft, and the vast majority of what happens is you get van der Waals interactions. Van der Waals interactions are hydrophilic, hydrophobic, pi-pi interactions, London forces, and that will then dictate the route by which the reaction goes. But it turns out that there's much more to it than that. There's much more to it. And that much more is that these are spin valves. They select out the electron with the proper spin. We don't use this in synthesis generally. We're just learning how to understand this. But this is how nature gets the selectivity that it gets. This is how nature does it. It's really, really extraordinary. Nature does this high selectivity by choosing the right electron. Scientists use pi conjugated molecules to get electron passage to occur through HOMO-LUMO gaps. I did this for a large part of my career. But cellular systems use primarily non-pi conjugated proteins for electron transfer. Researchers are just beginning to understand this. So theoretical work suggests that there can be several orders of magnitude enhancement of electron transfer in one direction over the other when going through chiral molecules. And this is what drives the selectivity. So might homochirality come after cellular life started? There's no evidence for claimed racemic mixtures where you have no homochirality being usable in cells. They have never been shown to function based on reaction specificities, yields, and heat management that would be needed for cellular systems. Vast numbers of electron transfers take place in lipid bilayer membranes, so it is baseless to summarily dismiss the need for lipid homochirality. How did the proteins noted above become homochiral when there's no prebiotically relevant route ever shown to make the amino acids in homochiral form, or to separate the enantiomers, or to polymerize them? But much like that which is promoted by those speaking about chemistries of life's origin, there are incorrect claims that make the chemistry of life appear easy, but it's not easy at all. All right, then you talk about the assembly. Once you make these molecules, you have to think about assembling them. A protocell is a self-organized, endogenously ordered spherical collection of lipids proposed as a stepping stone to the origin of life. Most so-called protocell assembly experiments in origin of life can be summed up by a protocol like this. You buy a homochiral diacetyl lipid from a chemical company or synthesize stereochemically scrambled lipids from smaller molecules. Add those to water and watch them arrange thermodynamically driven assembly into these bilayers. You publish a paper claiming that the synthetic vesicle is a protocell and suggestive of early forms of life. You engage with the media to ramp up the hype, and you watch the layperson be misled. Happens all the time. These lipid bilayers are super complex. And you say, well, the cells were simpler back then. We'll get to that. All right, so what happens is the outer part of this cell assembly is different than the inner part. And it has to be to maintain a proton gradient. You have to maintain a proton gradient across this thing. So how you get the outside different than the inside is absolutely unknown. We can do it with enzymes. There are flipase enzymes that can do this. But on a prebiotic Earth, how is that known? We have no idea. Then you have to have transmembrane proteins that allow the passage of certain ions through and other ions not through. You have to have transmembrane proteins that let other things through. You have these glycans coming off, these sugars molecules coming off, and you have to be able to have that for cell recognition. And so there's so much going on here, so much that has to happen in this. All right, how about the interactomes? Again, we're just learning about interactomes. It's the non-covalent interactive connectivity within a functional cell. Nobody knows how a viable cell emerged from the massive combinatorial complexity of its molecular components. And of course, nobody has ever synthetically mimicked it. An interactome is the whole set of molecular interactions in a particular cell. The non-covalent. If one merely considers just the protein-protein, we're not talking about protein DNA, a protein saccharide, just protein-protein interactome in just a single yeast cell, a simple cell of yeast, the result is an estimated 10 to the 79 billion combinations. Now that's a big number. Give you an idea of how big that is there's an estimated 10 to the 90th number of elemental particles in the universe. So when I say elemental particles, am I talking about elements or am I talking about electrons, protons, and neutrons? It really doesn't matter, because the difference between the element and the number of electrons, protons, and neutrons that make it up might be a factor of 100. A factor of 100 doesn't mean anything when you're talking about 10 to the 90th. When you're talking about 10 to the 79 billion, this is a crazy big number. And this is the non-covalent interactions that need to take place to have these cells operating. And that's why you can't take a cell and dehydrate it completely, where you remove the structural waters, and ever rehydrate it and make the cell again, because you've lost these. And that's why when a cell will replicate, what it'll do is will pass information to both sides, and then it'll cram down and split apart. So all of that information on how this thing these interactomes are put together are stored in that. So it just keeps passing down. Then there's the origin of information. Critical for life is the origin of information, DNA or RNA. The information is primary, the matter is secondary. but we can't even get the matter, the requisite carbohydrates, nucleic acids, lipids, and proteins, let alone the information. Let me give you an example of what I mean that the information is primary, the matter is secondary. If I have a thought, there's some information there, and now I take that thought in my mind, I take that thought, and now that becomes now stored in in first there'll be electronic storage, then there's protein synthesis, and then there's direct hardwired interconnections that are going to take place, usually within hours of that, that will begin to really bring that into a hardwired like memory. But now I take that thought and I write it down on a piece of paper. All right, so now it's gone from my brain to a piece of paper, two different types of matter, the same thought is written upon that. Now I take that piece of paper and I type it into my computer. All right, so that information has gone now from that piece of paper in my brain to go into key hits that are going to have that same information. That's going to go into some pressure upon a keypad. So the information is there. Then that's going to transfer electronically. Actually, this keyboard that I have is wireless, so it transfers through an RF wave to a little detector, and that then goes into a DRAM, SRAM, so DRAM, SRAM, and then flash memory into my computer when I hit save. So it's gone into 3 different transistor types. And now I want to save that to the cloud. So I save it to the cloud, so it goes through a wire and then goes into a modem and then goes through an RF wave into a box on the wall. So now that same information now is in an RF wave, and that's now in a box on the wall, and that goes back into a wire, and then through a wire it goes into a server farm and dumps into another transistor. You see all those places of matter that information was upon? But it's the same core information. The information is the same. The matter has changed. The information is primary, the matter is secondary. We don't even know how to make the matter, the 4 classes of compounds, in a prebiotically relevant manner. If you say, well, they came here from another planet, that's fine. I'm okay with that. We're talking about the origin of first life. We're talking about the origin of these things. But more important than even the matter upon which it is stored is the information itself. In the DNA, in the RNA, is prescribed all of this information. Where did that come from? We don't know. Try to build a cell even hypothetically. So bring together a dream team. A dream team can't make a living cell if given all the chemicals in homochiral form and the informational code. So even if you gave them all the chemicals and everything, and you even gave them the code, say build a cell, they can't. Nobody has ever even built a simple cell today. Nobody's done it, even if they took all the components from living systems to go ahead and make a cell. So just so that we're clear on this, in 2010, Craig Venter's group copied an existing bacterial genome and transplanted it into another cell. In 2016, Venter Group did something similar by removing all but 473 genes from a natural genome and transplanted it back into another cell. But you see, he already had a cell. He just knocked out all these pieces. Then in 2021, he added 19 back into those 473 because that cell didn't reproduce very well. So he added 19 back, and so then it could reproduce. So that's like starting with a car that runs beautifully. You go out, buy a new car. Go out, buy a Mercedes, and then you just start breaking parts until that car can start, but it can't move. That's what he did when he knocked out 473 of these things. And so then he went back and he added some parts back in until it started moving. That isn't making a cell. He started with a car already. Or when he took the genome out of one cell and put it into another cell, that wasn't making the cell. He took an existing genome and he put it into another cell, or he copied this genome and he put it into another cell. That's like saying, I'll copy this engine, I'll put it in that car. Did I make the car? I mean, the car already existed. So so that in case anybody wants to bring to me these synthetic cell experiments. All right, so a challenge, make the simplest living cell. A Nobel Prize would be awarded if anyone could make a working cell. Not a complex cell, even just a simple cell, a minimal cell that shows the functions of life. So reminder, what are the characteristics of life? You have to have responsiveness to the environment. You have to have growth and change. You have to have the ability to reproduce. You have to have a metabolism and breathe, maintain homeostasis, and being made of cells and passing those traits on to offspring. All right? So people will say, oh, well, the cells were much simpler back then. Well, how simple were they? Let's check this. How simple were they? So this has been modeled. These people have modeled, so biophysicists can model what is the simplest cell there could be. What is the minimal cell requirements? What they come up with is there needs to be some form of metabolism to provide the molecular building blocks and energy necessary for synthesizing the cellular components. There has to be genetic replication from a template or an equivalent information processing and transfer machinery, and a boundary membrane that separates the cell from its environment. So they say minimally that's what it has to have. The necessity of coordination between boundary fission and full segregation of the previously generated twin genetic templates could be added to this definition. Another fundamental characteristic that could be added to this essential features of a minimal cell is the ability to evolve, which is a universal characteristic among all living cells. And we will make these latter two optional for this challenge. So for this challenge, we just require 1, 2, and 3. All right, so what they say is the smallest natural genome capable of autonomous growth or laboratory cultivation in pure culture and also in a defined medium is one of M. genitalium with 580 kilobases, 580,000 bases, or 580,000 nucleotides are needed for this. All right, that's a lot. That's for a minimal cell today. What they estimate is the first theoretical minimal gene set was proposed by Mushkin and Kunin and consisted of 256 genes. Later, one integrative study utilized a larger data set, including results from both experimental and computational approaches for the minimal genome, and predicted a set of 206 genes for a theoretical minimal gene set. 206 genes. We're not talking about nucleotides. We're talking about genes. What is a gene? It's a distinct sequence of nucleotides forming a part of a chromosome, the order of which determines the order of monomers in a polypeptide or nucleic acid molecule, which a cell or virus may synthesize. All right, so you're going to have at least 206 genes. So you can't just keep throwing this out. Oh, well, they were much simpler, Well, we calculated that. Not me. Others have calculated this. 206 genes are what you need. All right, so this is what the minimal gene set would have to do. Again, this is not my calculation. This is their calculation. This is what you'll have to be able to do. DNA replication, repair, restriction, and modification. A basic transcription machinery. Amino-acyl tRNA synthesis. tRNA maturation and modification. Ribosomal proteins, ribosomal function, maturation and modification. Translation factors, RNA degradation. Protein processing, folding, and secretion. Cellular division, transport, and energetic and intermediary metabolism, glycolysis, proton motive force generation, pentose phosphate pathway, lipid metabolism, and biosynthesis of nucleotides and cofactors. The authors did not include rRNA or tRNA genes, and they recognized that these basic substrate transport machinery could not be clearly defined, even though this minimal cell would rely greatly on the import of several substrates, including all 20 amino acids, for which it had no biosynthetic ability. So you'd have to be able to provide it with all 20 amino acids, in homochiral form, by the way, to this system to get it to operate. But that's minimally what you would need. I don't care if you say, well, we're not dealing with prokaryotes, we're dealing with eukaryotes. You're fine. You want to deal with a bacterium? Fine. Bacteria need this as well. This is what you need in a bacterium. So let's make the simplest living cell. Let's try to do this. Okay, so first of all, what you've got to do is you got to make these, the building blocks of the building blocks. You got to be able to make sugars. Remember, this has already 4 stereogenic centers plus this one, which is the glycosidic site, the anomeric carbon. So this can be up or down. The amino acids, they almost all have at least one chiral stereogenic center. The nucleotides again are going to, you have to solve the sugar problem to solve the nucleotide problem. And then you want to make the lipids. You have to be able to separate between the enantiotopic hydroxymethyl groups, this desymmetrization. All right, well, if it's too hard to make these, what we'll do is we can just give you all that. I'll provide all of this to you. How's that? All you got to do is polymerize these and make this this lipid. Well, try putting these together. We have no idea how this could have been done on a prebiotic earth. Peptides, no idea how these could have been strung together. These don't form in water, and even if you did have ability to form these, if you're in some hydrophobic area, how'd you deal with the side chains? Nobody knows. Nucleic acids don't just polymerize together spontaneously, because what happens is if you get this DNA, what happens is they don't get very big because they're condensation polymerizations. You have to keep things really pure. If you have RNA, this thing is going to spontaneously be decomposing again. So it's hard to get these things to properly polymerize. You have to activate this, and you'd have to activate these. So this would be RNA, and so you'd have the other OH here. And then you'd have to make the lipids here. Again, a big problem. How do you desymmetrize this? But if you had this lipids, again, you'd have to have this ethanolamine unit on there. All of this chemistry is hard, but I'll give you that too. I'll give it to you. You can have that. You can have all of these in polymerized form in any sequence you want. So when I give you sequence, I'm giving you the information as well. So you got the monomers, you got the polymers, you got the informational code. Nobel Prize is right there. All you got to do is make a living cell. You receive a Nobel Prize. You would be an instant rock star. I mean, you'd just be amazing if you did this. Well, you can't do it? Come on, a Nobel Prize awaits you. What's wrong? You're so close to making life in a lab. I can't understand what inhibits you. A mindless early Earth was able to do this, even though it had to make all the small molecules in homochiral form, and the polymers of those molecules, and the proton gradient inducing bilayers, and the interactomes, and everything else while working under a rock. You can use your entire lab and all the separated gases and any chemicals in pure form. Come on, you're better than a mindless Earth. That summarizes the state of assembling a cell. It cannot be done even if given all the pieces and the informational code. I'm not saying that it could never be done. As a scientist, I cannot know that, so I would not claim it. But I can say that we are nowhere close to solving this problem. How do I know that we are nowhere close? Because we can track progress relative to the target of making a living cell. And the goalpost keeps moving. And what I mean by that is that the complexity of a cell. So when Miller-Urey did their experiments in the 1950s, people thought that they were going to really solve this origin of life problem. But what happened is the complexity of the cell got greater and greater. So every year we learn more about the cell, like chiral-induced spin selectivity, like interactomes, and we go, yikes, it's a lot more complex than I thought. So the goalpost moves further away. And so the reason we know we're nowhere close to solving this is because it's not like we're getting closer and closer to the solution. We may be moving closer, but the solution is moving away at a faster than we're getting closer to it. That's the problem. We're nowhere close to solving this. So the next time you see an article like this that says researchers solve puzzle of origin of life, you just got to read the article. You got to go back to the article, the research article. And what you'd see is they just bought a bunch of building blocks and they stuck it in a lipid bilayer, and they say, wow, that shows the origin of the first cell because these things stick in the lipid bilayer. So, and you find that they just purchased all the building blocks and allowed them to associate at high concentrations in the lipid bilayer. It's far from a working cell or solving the puzzle of the origin of life on Earth. They've just bought a bunch of parts and they shoved it in here, and they thought it, wow, that must be a working vehicle. No, that's not a working vehicle. So I am not preaching God of the gaps, not at all. Just because I don't know how to do it doesn't mean that it's not going to be done. As a scientist, I would never say that we will never understand. One day in the distant future, we might understand life's origin and evolution of a complex system, but as of today, we just don't. And we remain clueless, and the problem gets harder all the time. If we do find out one day, that's not going to lessen God, but we'll see him as all the more magnanimous. So I'm not trying to preach God here, that's God of the gap, since we don't understand, it must be God. Please don't suggest that I'm doing that, because I'm not. I'm not suggesting a God of the gaps argument. I'm just saying that we are clueless on this. One day we might know, but that day is far from today. And with that, let me just bring this up. This is my if you want to write to me, you can write to me at drjamestour.org, drjamestour.org. And you can connect with me. I have a YouTube channel, Dr. James Tour, and I am a. I'll say right up front, right now, I am a person of faith. I didn't have to use any explanations of God or anything. No religion was needed to present this critique on the origin of life. But if you want to hear why I am a person who believes in the physical resurrection of Jesus Christ, you email me and tell me you want to hear my story. If you do not already believe in the physical resurrection of Jesus Christ, and you're interested in hearing my story about how I came from a Jewish background, and I believe that Jesus Christ is risen from the dead, you can email me, we'll get together, and I'll share with you my story. But that's only for people who do not already believe that Jesus Christ is risen from the dead. All right, with that, I'm going to end, and I'll open it up for questions.
Thank you so much, Professor Tua, for an amazing presentation. It was really kept me personally at the edge of my seat. I'd like to now, please, for participants who would like to ask Professor Tua, can you please raise your hands? Okay, let me quickly look at the list. We have the next person.
Niket Kumar.
Oh, he already asked earlier, Niket. Can we get someone else, please? Can we get the next person to be free?
Yes.
Thank you. I've asked you to unmute. Can you please unmute yourself?
I think he is Mr. Pramod.
Yes, sir.
Yeah.
This is Dr. Pramod speaking from Hyderabad. I'm a retired associate professor from University of Delhi. And thank you, Professor James. You have given a wonderful talk regarding the creation of the life. My question is that, as per the scriptures, Vedic science says that there are 84 lakhs of species. Do you agree with the statement? I can repeat the question. Vedic.
Yeah, I didn't understand your question, sir. I'm sorry.
The Vedic science, the scriptures say, Indian scripture says that there are 84 lakhs of species, as per our Bhagavad Gita, Vedic literature says. What does the science say about this?
So your Indian scriptures say that there are 84 species, and you want to know
84 lakhs. 84 lakhs. You see, 84 millions.
8.4 million. Can I just interject? 8.4 million.
Yes.
8.4 million species. And you want to know what science says about that?
Yes.
I don't know how many species science says there are. You'd have to speak to a biologist. I'm sorry, I just don't know. So I can't testify whether your scriptures are aligned with current science, but what I can tell you is that your scriptures are going to remain the same, but science is all over the map with its prediction of things. Science predicts all sorts of things, and then they keep revising their numbers. So trying to align our scriptures with scientific claims that are not facts but speculations is really hard, because science keeps changing with time. Your scriptures are going to remain the same. That's the best I can do, sir.
Thank you, sir.
You're welcome. Aristotle, you're muted.
We have one more question by Ms. Dr. Ramji Redka, please.
Yeah, thank you, Professor Teams. It was a nice presentation indeed. I have no question, rather I have a comment. You have nicely elucidated that one cannot create life or cell in the laboratory. And this is what Professor Dr. T.D. Singh, the founder president of this Bhaktivedanta Institute. So he has put the same question during his PhD time to Stanley Miller, that, okay, we will give all these elements. So can you bring life to that? And then the whole hall remains silent, including Stanley Miller. So, I
Good.
mean, I could get reminded by seeing your slide. It was very nicely presented. And then you have shown all the 4+4, all the building blocks. Though you can have at your hand, one can have at their hand, but it is impossible for them to bring simple living cell. Thank you so much. Thank you so much. And you
Oh,
have shown from biology background. Very nice. Thank you so much.
thank you.
Of course, in
Thank
the
you.
form of chemical, I mean, chemical molecules. Thank you so much.
Thank you.
Thank you. Can I ask Mr. Vasudev Rao to please ask the next question?
Thank you, Professor James, Dr. James. It was a really wonderful, eye-opening lecture.
Yeah, Vasudev Raoji, your audio is a bit low.
Just a minute. How is it now?
Yeah,
Is good enough?
little bit increased, but some kind of disturbance is coming.
Okay. So, thank you very much, James Tour. It is a fantastic lecture, very eye-opening. My question is, from what you have presented in your lecture, and what generally is perceived within the scientific circles, and what is presented to the students, the state of the art about the origin of life research, there appears to be a big gap. So, how to bridge this gap? We definitely some kind of exaggeration, little exaggeration is good to encourage research and the direction some of the prominent scientists are seeing. At the same time, it is also important to present reasonably the state of the art. So, how this gap can be built?
Is it clear,
Aristotle,
Mr.?
Aristotle, can you repeat the question?
His voice was quite unclear, and I was actually typing a message to someone.
Okay, just a minute.
Can you say that again, please? Just the last part of your question, Mr. Vasudev.
Just a minute.
Hello. Is good enough now?
Yeah.
Very good now. Very good now.
So my question is that from your presentation, we see there is a big gap between the state of the research in origin of life, or how first living cell originated, and what is believed in the scientific circles, or what is presented to the students of science. There is a big gap. So while some little exaggeration is good to excite in the science and scientific research, but it is also necessary to present reasonably the state of research, so that the alternative ways, alternative paradigms, they get more life by the researchers and the students. So how to build this gap? There is a big gap.
Yes. You know, I absolutely agree with you. So what happens is we teach our students a bunch of nonsense. Your last speaker said something really quite profound, that the science has become politicized. And if one says what I just told you today, if one says this in the United States, they're not going to get tenure. They're not going to get promoted. I can say this because I already have tenure. and I'm already in a position that they can't do much to me in my career. If they don't want to give me certain awards or something, that's fine. And I still am, I'm pushing the bounds of science, but it's very hard to advance if you say these things because you're pushing politically against the system. It's hard to have a Galileo today. The same things that happened to Galileo will happen today to people if they say these sorts of things. I think it's very important, I agree with you, to really tell them the state of how bad things are in our knowledge on origin of life. I've even gone so far as to say there should be a moratorium on origin of life research the way it's practiced. And we should have a real change. We should get together and say, what are the targets? What do we have to show here? So that we can get a lot of young people to say, okay, let me come with totally different paradigms on this. DARPA, the Defense Advanced Research Projects Agency in the US, they have programs like this for the military. They'll say, okay, nobody's solved this problem. We've tried for years. So we're only going to take proposals that are totally out of the box, totally new. And they stimulate young people to contribute. They don't want the old guard people to keep saying the same old thing. They want only totally new ideas. I think we need that in Origin of Life because we're so clueless on this and we keep propagating this nonsense. And I'll tell you, people have disagreed with me, but this is taught. I have slides on textbooks from high school and from college and from graduate school that still teach this prebiotic pool where there was lightning strikes on a pool and some molecules formed and those got together and formed cells and those cells formed slithering organisms and they came out came life. This prebiotic pool thing is still taught even at the graduate level in biology textbooks. And so students think, oh, this is how it happened. No, we have to tell them that's not how, that's clueless. We're clueless on how that could happen like that. And we have to give them the freedom to say, maybe they'll come up with some great ideas on this thing. But it's a lonely and it's a hard world to be a voice crying in the wilderness that we're clueless and your experiments don't show us anything on how origin of life could have done because it's prebiotically irrelevant. And I've taken a lot of hits on the internet for this and they continue to come at me. So yeah, science has been politicized and when you come against the status quo, and I'm not sure what the origin, because I'm not a philosopher, what is the origin of this that makes people so upset about just saying we don't know? I don't know. But anyway, I agree with you, sir.
Thank you. Can we have Niket? Niket, can you please unmute yourself?
Yes, sir. Yes, you can hear me, no?
Yes, very clearly.
Yes, sir. Thank you for giving the opportunity to ask him questions, and thank you for the wonderful organic chemistry lecture also on origin of life. My question, can I ask? Can I start asking?
Yes, please go ahead.
Yes. Reagent addition to prebiotic system synthesis was told by him. So means it meant that if even if some reagent kind of substances is added to the prebiotic system, it starts stimulating life. So in that way also somehow the theory is looking good, no, that there is some kind of might be some prebiotic pool. So some means like earth was very hot earlier, then after the rain came, then in pond some microbes start means arising there, like some reagent kind of thing. So in that way, like, is it not validating the reagent addition to prebiotic system in that way?
No, it's a
That is
bunch of
the
nonsense.
question.
No, this is nonsense. This is what they say. This reagent comes in and it stimulates this. It doesn't. We have no idea of the chemistry that's needed. People will only say this when they're not held to account of the organic chemistry that needs to take place. As soon as you ask them for the organic chemistry details, everything utterly falls apart. That is utter nonsense that you've been taught, and you are a product of this propagation of propaganda that is going out on the origin of life. You're a product of that, sadly. So that's a bunch of nonsense. And there was
Yes,
a
sir. So
comment,
like,
there was a comment that came up in the chat that Craig Venter made the first living cell, and I showed Craig Venter's work. I discussed that, and he did not make a living cell. He did not take a cell ab initio. He took a cell, he knocked some things out, and he said it was a synthetic cell. It was a poorly working cell because he took a good working one and he knocked stuff out of it. And it was so bad that he now put some more stuff back into it, got it working a little bit better, but he just made it a little bit better from the bad state that he had set it to. But I addressed that in my seminar
yes,
already.
sir.
Yeah, I think Professor Chu had already talked about the building blocks of the building blocks as well as the building blocks, and even if they got all of it, they wouldn't be able to put it together. We have another
That's right.
question by Suman. Suman, please unmute yourself.
Hello, hello, sir.
Hello.
Hello, sir. I'm a researcher in molecular biology. So I want to ask you a question that do you think that the RNA world hypothesis is exactly true, or is there any concrete evidence on that RNA world hypothesis that is very much popular in origin of life theory?
Yeah, it's been questioned from the time it's come forth, and it's still being highly questioned. The problem with RNA is it tends to form duplexes that are hard to pull apart. It's the sticky problem. The other thing is it's unstable. The stuff is unstable. You're building a whole hypothesis based on something that is unstable. I mean, at least DNA is stable. It doesn't undergo this autocatalysis, this autodegradation. RNA, it decomposes. And then if you try to make it, if you try to make RNA, you get not just the 3-5 linkage, you get the 2-5 linkage. And the 2-5 linkages act as siRNA, small interfering RNA. So it stops the process, it shuts the thing down. And if you say, well, it was made upon clay, well, you look at the experiments where they made it upon clay. They had to add ion exchange resins and acrylonitrile in order to get it off because it sticks like crazy to the clay because clay is positively charged, RNA is negatively charged, and if it ever got on the clay, it would never come off the clay. Now, where are you going to get an ion exchange resin on early Earth and not acrylonitrile, but acetonitrile to pull this thing off? It's just a hard problem. And if you look at the stuff that they made, there was no particular order to it, and it was full of 2-5 linkages. And when they make these claims, they never go into the details. You get 30 to 80%. 30 to 80% of the linkages are 2-5 and not 3-5. And that acts as siRNA. So I think the RNA world hypothesis is a bunch of garbage. It is garbage. It makes no organic chemical sense. You're making an unstable molecule. It sticks to itself. It sticks to other things such that it doesn't come off. And it falls apart very rapidly. And a lot of what's made argues against the RNA ever working for anything. Also, this whole thing that RNA duplicates itself is a lie. Even the experiments, when you look at Gerald Joyce's experiments, who's a great scientist, but you look at his experiments, he used primed RNA that's designed to duplicate itself. You know how much duplicated itself? Less than 10%. Less than 10% of the RNA strand was able to duplicate itself. That's not duplication. That's only 10%, and that 10% was too short to duplicate any more of itself. So this whole thing that RNA, all you need is a strand of RNA and starts duplicating itself and it's a catalyst and goes on. Nonsense, utter nonsense. Show me in a laboratory. They won't, because it doesn't work. It's all mind confined.
Okay, sir, I want
That's
to
what I
ask,
think of.
sir, I want to know which hypothesis, according to you, is more relevant, like glycine world hypothesis or peptide world hypothesis. Which hypothesis on we can rely? So
I have no idea, and some people tell me that I shouldn't criticize other ideas until I have my own idea. That is totally unlike how we do science. Within the realms of organic chemistry, if we see a mechanism that doesn't meet up with the facts that we have in the laboratory, we discard that mechanism. We don't say, oh, I can't discard that mechanism. Even though I know it's wrong, I can't discard it until I have a new one. I am telling you, I am clueless scientifically on the origin of life. I'm clueless. None
thank
of
you,
these
sir.
mechanisms make any good sense to me.
Thank you, sir.
We have 3 more questions, very quick questions. Please keep your questions succinct. We have first Mr. Warren Agarwal, and then followed by Dr. Jain, Dr. Jayanti.
Okay, James, a great lecture, remarkable lecture and very bold lecture. And we are very excited and energized by your presentation, very straightforward. I have a small quick question is that, one of the arguments, one of the escape path many times is used is that for any questions asked about the origin of life, is that well, given a long period of time these things can happen, right?
Right.
Given a billion years of time, this can
Yes.
happen or that can happen, and that becomes like an escape path for anything. So one of the proposal is that one of the thought was that could we to avoid this escape path, or are there possibility of having a super some kind of catalyst so that this path also can be clarified? Means you we try to have some kind of super catalyst which brings the things together and we can see the result in a short time. Means there is no room for doubt remains. Means you got my thought point. I think you
I
got
got
some
your
idea.
point. All right, let me just tell you, time is not a solution, it's a problem. I have showed this over and over again using the exact data from these origin of life researchers, where you see that they get a certain yield after a few hours and then they stop the reaction like any good organic chemist does. Because when you don't stop the reaction, then you see what you want decompose. Generally on these reactions, that's not the thermodynamic product. I mean, there are other polymerization products. You do a condensation reaction to the foremost reaction to try to get you carbohydrates. I mean, these things go to polymers very quickly. You got to stop it. And then even when you get it under the very reactions that made them, they undergo decomposition. So time is the enemy. We know time is the enemy for organic compounds because they undergo oxidation. I mean, to ask any organic chemist, do you leave your chemicals out on the bench just for days and days and days and months? No, you get oxidation. These things decompose. If you say, well, the early earth wasn't oxidizing, it was reducing. Oh, really? Tell me what what ammonia does to molecules. Put your molecules under an ammonia atmosphere and go away for a little while and see what happens. And so these things degrade. And then you have light and they'll shine UV light in order to get reactions to go, but then they turn off the UV light right away. They don't want UV light hitting the molecules they want. So it's a bunch of nonsense. A super catalyst, look, catalysts are extremely selective. If you perchance had a great catalyst, it would do one reaction. It doesn't do everything. It doesn't do all reactions. We know of no such super catalyst. So you're pulling things, based on faith. If you want to do things based on faith, we might as well just say, well, God created the heavens and the earth. That's it. Let's go home. He did it. Then he's our super catalyst. So you'd need a super catalyst like God to do exactly what you're saying if you wanted to catalyze all of these reactions, all the different types of reactions in a short amount of time.
Oh,
Can we have the next question, please, by Dr. Jain? 009.
thank you. Good. Thank you. Good thing. Thank you, James.
Okay.
Yes. Can you hear me?
Yes.
Yes. Thank you, sir. I'm Dr. Jayanti from Institute of Science and Religion. Your presentation was wonderful. It was very compact and descriptive and explorative. And I have a variation question from what Sri Varun Agarwal just asked. That from your presentation, it is very clear that it is very complex to understand life's origin and as well as the evolution, as well as complex system. But you're optimist. You're optimist, and you said that as a scientist, I would never say that we will never understand. And one day in distant future, we might understand the life's origin and evolution. So what is that? Giving you that confidence? Because you have already spent so many years in researching, and you had also been a main critic for this origin of life. So what is that that is bringing in the optimism in you that you will be able to understand this origin of life in the future?
Yeah, I'll tell you, if somebody asked a researcher in, say, during the Puritan times, say in the 1700s, why is it that when the parents are tall, their children are tall. When the parents are short, the children are short. All they'd be able to say, well, you know, it must be God. I mean, I have no idea. You know, we now know from the 1950s about DNA, about how information is stored. And that opened up so much to us in the understanding of cellular operation, of passing information down, of these sorts of things. So there are discoveries that just bring huge enlightenment to us. But if you ask somebody 100 years before that discovery, they're absolutely clueless. We are in an absolutely clueless state. So as a scientist, I can never say we will never know because I can't predict the future. And I presume that we will know, but again, it's a presumption. You think I'm a great optimist? I receive that. That's wonderful. Most people think I am the most pessimistic person in the world on this because they feel that they've already got it solved. And but you think I'm a great optimist. That's great. So I think that human discovery is amazing, and they keep coming up with amazing things. I mean, maybe we'll never know, but I think, you know, we'll have some ideas. And then when we learn it, so when we learned about DNA and how this information was stored, it did not make us angry at God. Oh, why now we know. No, it made us like, wow, God, you're amazing. What an amazing way to store information. You store information, it's a stable molecule. You got each one of three of these encodes a particular protein. The proteins then get built from this, and they are the little nanomachines that assemble the structure within a cell. Wow, what an architecture. And so I think as a scientist, it makes me love God all the more when we see these advances. So anyway, I answered a little bit more than what you
Yeah.
asked, but
Yes, sir. But
hopefully
my
my
question is, I understand that because when Francis Collin has also seen the God in the DNA, but now considering the history back, it has been only unfolding of living, non-living system, non-living DNA molecule. But it was never about life, which you said the characteristics, and never it has happened in the past history that it could be known. And whatever the history on the back is not connected at all to the characteristics of life. So what makes a scientist think that they will be able to understand the origin of life? Because the
well,
history has
I guess I'm optimistic. You're right. I'm optimistic. I mean, I don't have a lot of scientific basis for that optimism.
Thank you,
Just
Dr. James.
thank you, thank you so much.
inherently.
James, Professor James. We have one last question by Sushma, 0018. Can you please unmute yourself? Hi, Sushma.
Hello, very profound, James Tour, very profound lecture. I have a quick question, like how these people, like how Sri Varun was asking that these people, they take shelter of time, but many times we, on this side, we also take shelter of probability. And we take that it is so highly improbable, 1 to the power of this. And but that number is still not a zero number. It is still not zero. So like how they justify their defense, how do you think we should justify our defense? Thank you very much.
You know, I didn't use, you know, a lot of people use mathematical arguments a lot. The only thing I used was within the context of the synthetic chemistry. When I said it's just hard to put together carbohydrates because of all the different ways you can put these things together. Then when you go to interactomes and you have numbers like 10 to the 79 billion, you know, this is a crazy big number. You know that when people say 10 to the 40th, that's essentially never going to happen. When you go to the 10 to the 79 billion and you go, wow, these are hard numbers. Again, that's why I come out at the end. I don't say we will never know. You know, I can't say that. I just say my whole argument is that we are presently clueless on the origin of life. We're clueless on this. This argument that you're saying, it's the physicists mainly that come with this fine-tuning argument. I didn't use any of the fine-tuning argument. I just use what I deal with as an organic chemist. And for any of you, I know Indians love organic chemistry. Many of them do. When you do organic chemistry, you don't do a lot of mathematics. You just get in the lab and you build things. But there are probabilities you have to deal with. I mean, when you're numbers like this, it's just staggeringly small. Their time is actually very short. Even if you go with this, we know that the Earth was pelted by big meteorites just pelting the Earth. And we know that the Earth got pelted 6 times more than the moon did just because of our mass. And the Earth heated, and any bit of life would have been burned up. From the time that the Earth is presumed to have cooled till the time there is life, we are talking about things on the order of hundreds of thousands of years. So even the time argument for them goes away, because we can see early cellular life, the fossils of early cellular life that had to be there from as soon as the Earth cooled. As soon as the Earth cooled, we see cellular life. And those cells that we see were as complex as our simplest cells today. We don't see transitional cells. We see the cells that we find in the fossil record appear as if they were as complex as our simplest cells today. And so they appeared immediately. So even their argument of time now goes away, because you can't take time from the beginning of the universe if you're talking about on this Earth, because whatever was on this Earth burned up. Now, if it came from another place, okay, but again, we're going to have to then say, how did it form over there? But even if you go with the 14 billion years, that's not very long when you're dealing with numbers like what people show that you have to contest with on origin of life. When you're talking about just six carbohydrates, it's just 6 carbohydrates stuck together, has over 1 trillion combinations. This is going to go up exponentially very fast. So I think if you take the numbers game, we win hands down.
Mhm. Thank you very much. By the way, I forgot to introduce myself. I'm Sushant, patron from Bhaktivedanta Institute. Thank you very much.
And thank you, Professor James Truer, for a very riveting discussion and presentation. Thank you so much.
Well, let me just say, you've been a very engaging audience. I think you guys are more excited about this type of thing than Americans are, because anyway, you've been really, really engaging. I appreciate the invitation. This has been a joy for me. I mean, to think that so many people would be so engaged on this topic. You made me very happy. Thank you.
There are many more questions actually, but because of time, we are unable to take them right now.
Yeah.
Thank you.
Okay. Bye-bye.
So let's give a round of applause to Professor James Truer for an amazing presentation.
Thank you.
Thank you
Bye.
so much for blessing us.